There are 8 billion people on the planet, and all of us are wonderfully unique. We have different eye color, height, shape of mouth and nose, and dramatically different way of thinking. But, at the same time, there are characteristics that we all share. Wherever you go, you will find people who walk, breathe, and digest food the same way. Where do these diversity and similarity come from? Human body is a collection of 30 to 40 trillion cells. Each of these cells contains a nucleus, and inside each nucleus is your genome, your DNA, that carries your genetic information. Most of the cell's structures and operational systems are made from a variety of proteins. DNA is an encoded instruction, digital information, on how to build proteins. This genetic code is read by the cell and put into practice. It specifies both the features we all share and the inherited characteristics that make us different to one another. In other words, genome is the software that codes for you. It's your hair color, a significant part of your personality, and even susceptibility to specific diseases. Think about how your computers and smartphones work. Any form of digital information is essentially a series of 2 letters, 0 and 1. Your devices read this binary code of information and turns it into a picture, a video, a spreadsheet, or whatever, and show it on the screen. Similarly, our DNA consists of 4 types of letters, adenine, thymine, guanine and cytosine, or ATGC for short. The cell reads this quaternary code to build a protein, and the protein realizes the form or function of the organism. Genome has 3.2 billion letters of DNA. If it were a book with a standard size font, it would have 1.5 million pages, full of just 4 letters, 60 times the size of an ordinary encyclopedia. These 3.2 billion letters are divided and packaged into 23 pairs of chromosomes, in a double helix form. But, the vast majority has nothing to do with your genetic information. Only about 2% of DNA encode proteins, which we call genes. Human genome contains about 22,000 genes in total, only 1,800 more than worms, and 25,000 fewer than rice or wheat. There is nothing particularly special about human genome. Another critical function of DNA is its ability to copy itself very precisely. Most of the genetic code for the various cellular components are the same in all organisms, from plants to animals, tiny bacteria to Homo sapiens. That means, the core information in those genes has been preserved for probably 3 billion years. DNA can replicate and preserve itself with very few errors. Then how does DNA's structure make it possible? DNA contains 2 chains. Each chain is a long sequence of 4 bases, adenine, thymine, guanine and cytosine, or ATGC. The bases can only pair up in a single precise way. A can only pair with T, and G can only pair with C. One chain is a reflection of the other. This means that if you know the order of bases along one chain of DNA, you immediately know the order of the other. If you break the double helix apart into 2 separate chains, each chain can act as a template to recreate a perfect copy of its original partner chain. This is the way that cell division takes place, without almost no copy errors. If DNA is an encoded blueprint of the cell and the whole human body, how does each cell decipher these encrypted messages to build proteins? Let's go back to the analogy of DNA and digital devices, and see what happens when you text your friend hi. One letter on computers is encoded into 8-digit binary code. For example, capital H is represented as 01001000. When you text your friend hi, what you're actually sending is just a 16 digits of 0 and 1. Genetic code functions quite similarly. DNA consists of 4 bases, ATGC. And 3 bases come together to carry one piece of information. But why does it have to be 3-letter code? A protein is created from 20 simple chemicals, named amino acids. A quaternary code of single digit would only represent 4 amino acids. A 2-digit would code for 16. A triple digit of quaternary code can represent 64 messages, enough to express 20 kinds of amino acids, and how to control them. For example, ACT specifies the amino acid threonine, and ATG is the code to start the building of a protein. By the end of the 20th century, entire genomes could be sequenced. This was the goal of the Human Genome Project, a $100 million, 10-year effort. Since then, though, the price has plummeted. Today, sequencing a human genome takes a few days and costs less than $1,000. It was a major step forward for biology and for medicine. When you understand your genome, you will learn the diseases to which you're most susceptible, and more importantly, how to prevent and cure them. The genome contains the memory to build every cell, in every tissue, in every organism. All the cells in your body have the same collection of 3.2 billion DNA letters. The next question is, what distinguishes a brain cell from a liver cell, a skin cell from a bone marrow cell, even though they have the exact same set of DNA? When the cell reads its genetic information, DNA code is transcribed into a messenger RNA code. The RNA copy then moves from the nucleus to the cytosol, where its messages are decoded to build a protein. But the transcription to RNA is done selectively. Not all DNA is copied into RNA. The key to understand this mechanism is gene regulation, the set of chemical reactions which cells use to turn genes on and off. For example, the cells in your kidney, skin, and brain all contain the same total set of 22,000 genes. Gene regulation means the genes needed to make a kidney were turned on in embryonic kidney cells, and those that function specifically to create skin or brain were turned off, and vice versa. Once the growing embryo instructs cells to be kidney cells, they remember that information, and rarely change their identity. Once a cell is determined to be a kidney cell, it will remain part of the kidney, by turning off unnecessary information. This mechanism of turning genes on and off is called epigenetics. It does not change the DNA sequence of the genes themselves. Instead, it works by adding chemical tags to the DNA. These tags tell which genes should be on and off. Only parts of genes that are on are selectively transcribed into messenger RNA to build proteins.